Targeting Bruton tyrosine kinase has revolutionized treatment of chronic lymphocytic leukemia and certain subtypes of non-Hodgkin lymphoma. However, resistance to BTK inhibitors such as ibrutinib, acalabrutinib, and zanubrutinib emerges invariably, and it now has become an unmet medical need. Pirtobrutinib has recently been approved in treatment of CLL and mantle cell lymphoma, and it is also associated with resistance. So resistance occurs because of mutations in BTK, in the Bruton tyrosine kinase, and different mutation patterns are associated with different covalent and non-covalent BTK inhibitors. But nevertheless, all of this results in increased activation of BTK kinase, of BTK, the protein BTK kinase, and resistance to therapies, where these inhibitors can no longer disrupt its function. BTK degraders is a class of PROTACs, proteolysis targeted chimeras, which rely on degradation of BTK, where they don't necessarily care, these molecules don't necessarily care what type of mutation is present in BTK. NX-2127 is a BTK degrader which has been in clinical trials now for about a year and a half, maybe 2 years. In this study, we enrolled 47 patients with CLL and non-Hodgkin lymphoma, and the drug is given orally daily. The drug has been really well tolerated. The side effects are mainly typical of what we see with BTK inhibitors such as ibrutinib. We've seen some bruising, some neutropenia. However, there were no significant adverse events, and very few patients discontinued because of adverse events. For example, in a group of 29 CLL patients, the overall response rate was 40%. However, many more patients actually achieved clinical benefit, because responses in CLL can be quite slow. In patients with non-Hodgkin lymphoma, we have also seen responses. So this trial is ongoing, and this represents a completely novel mechanism which will enhance our armamentarium of treatment for patients with BTK inhibitor refractory disease.